The purpose of this video is to cover Chapter 6 for Bio 202, and the point of Chapter 6 is to introduce you to some basic concepts about energy and enzymes, and we'll talk about how they relate to metabolic reactions in the cell. So we'll be talking about both photosynthesis in Chapter 7 and cellular respiration in Chapter 8. These are big metabolic reactions that occur in the cell, and they require energy and enzymes to do so. In the course of this lecture, then, we'll talk about the big question of how enzymes facilitate metabolic reactions. To do that, we have to first understand what are the different forms of energy and how is energy converted from one form to the other. And to understand that, we'll be taking a look at the two laws of thermodynamics. From there, we'll look at how ATP plays a major role in metabolic reactions, and especially in the coupled reactions that take place in the cell. Enzymes play a critical role in metabolic pathways and metabolic reactions, so we'll take a look at how enzymes facilitate metabolic reactions and really focus on some of the factors that affect enzymatic reactions. We'll also have experience with this in one of our labs, upcoming labs. Overall, the idea is to understand how all of these things play a role in the energy organelles that function during photosynthesis, so that's the chloroplast, and also then during cellular respiration through the mitochondria, so how these different energy organelles facilitate the conversion of energy. So that's where we're headed in this lecture. Okay, so energy comes in many forms. It can be potential energy, kinetic energy. We can talk about solar energy or chemical energy or mechanical energy. So the whole idea of this chapter is to understand how we can convert energy from one form to the other, how the cell does that. And it basically takes some form of energy like solar energy and makes it into a usable form that the cell can use. This slide here is showing solar energy being converted to chemical energy. And specifically, this is through the process of photosynthesis, which the chemical energy in that case will be in the form of sugar or glucose. The animal here is then eating the leaves that now contain the glucose or chemical energy, and through the process of cellular respiration, will then convert it to mechanical energy, and we're going to talk about mechanical energy. The chemical energy being converted to ATP. ATP is then used to drive reactions such as muscle movement, and so that would be the mechanical energy. The first law of thermodynamics states that energy cannot be created or destroyed, but it can be changed from one form to another. In other words, that's what this slide is showing, the conversion of solar energy to chemical energy and the conversion of chemical energy to mechanical energy. So it's not created or destroyed, but changed from one form to another. This process is not 100% efficient, but we don't lose any energy in the process. Along the way, some of that energy is converted to heat. So not all of solar energy is completely converted to chemical energy, but some is basically lost as heat. But it's not destroyed. It's just converted to both chemical energy and heat or mechanical energy and heat. All right? The second law of thermodynamics is a little bit more complicated. It states that the amount of entropy in a closed system will increase. In other words, the conversion of energy will result in a loss of usable energy. That's basically what I just said. We have some energy that can be converted from sugar, stored chemical energy that sugars use to drive muscle contractions, but some of that energy is lost as heat. Heat is not usable in the cell, so basically it results in a loss of usable energy. In other words, the conversion is not 100% complete. So what is this idea of entropy? Entropy is a little bit more challenging to understand. Basically, entropy is energy not available for work. It's unusable energy. In this case, the heat, for example, would be a loss of usable energy. So when there's an increase in entropy, it means there's more disorder and less usable energy. But in reality, this is a much more stable situation. So let me try to explain this. Here we have glucose. Glucose is a very organized molecule. There's a lot of energy stored, potential energy stored in a glucose molecule, and it's pretty stable. There's not very much entropy. All right? Glucose will be broken down into carbon dioxide and water through the process of cellular respiration. This is much less organized. It has less potential energy. There's not as much energy stored in carbon dioxide and water as there is glucose, but it's much more stable. It's been broken down to its component parts, and it's much more stable. It has a high amount of entropy. Okay? It's more stable, but there's also a loss of usable energy because there's less energy stored in carbon dioxide and water. So the amount of entropy has increased in this process. So whenever there's a conversion of energy from one form like glucose to another, there's a loss of energy that increases entropy. It's much more stable. Here's another example. We've already talked about protein channels that allow ions to pass through the membrane. So here's an example of an ion channel that allows the movement of hydrogen ions. So here we have an unequal distribution. There's more hydrogen ions in one side of the membrane than the other. It's very organized in this fashion. It has more potential energy because that difference between the number of ions on one side of the membrane and the other represents a electrochemical potential. So here we have more potential energy. But it's not very stable. We talked about the idea of diffusion, how molecules want to move from a high concentration to a low concentration. So this situation is not very stable. This energy here, as ions move, is a conversion of energy. So here we have a conversion of energy. Some energy is lost. Now we have a much more stable situation here where there's an equal distribution of hydrogen ions, but it's much less organized because we have ions on both sides of the membrane. But it's much more stable, and it has a higher entropy. All right, so entropy increases the more stable something becomes, but it shows a less potential or stored energy. Think about your room. If you have a messy room, it's much more stable when it's messy, right? You can maintain a messy room. To get it back to a more organized state, you have to input energy. All right? When you have a very clean room, and it's much more stable to move to messy, but you lose some of that potential organization. And so entropy increases as it becomes more messy. So in general, reactions want to happen to become more stable, but it results in the loss of usable energy. So to sum up, in general, there's going to have to be an input of energy and a decrease in entropy to make something store energy. Then as reactions occur and there's an increase in entropy, there's going to be a release of energy as there's an energy conversion. Okay, so now let's take a look at metabolic reactions and how they relate to the ATP cycle. We've already talked about ATP being a nucleotide, and it is specifically adenosine triphosphate. All right, so here we have the sugar and the nitrogen-containing base, and as a triphosphate, it has 3 phosphates added to the sugar here. Remember that it takes a lot of energy to keep these phosphates attached to one another, so there's a lot of energy in those bonds. It has a lot of stored energy. As it loses those phosphates, it's going to release energy. It's going to become much more stable when it becomes a DP, or diphosphate, and it loses one of those phosphates. It's going to release energy. Reactions that release energy are called exergonic reactions. So when ATP is converted to ADP. It's an exergonic reaction that releases energy. Reactions that require energy, an input of energy, are called endergonic reactions. So here we can couple two reactions together, one reaction that releases energy and one reaction that requires energy. You can couple them together. The one that's releasing energy can provide the energy necessary to drive the endergonic reaction. So in this case, ATP releasing energy can be used to drive an endergonic reaction. Things like muscle contraction that require an input of energy. Protein synthesis requires energy. Nerve conduction requires energy. ATP, as the energy currency of the cell, can provide the energy necessary to drive endergonic reactions. And when it does that, it goes from ATP to ADP. ADP has to then be rebuilt up in the cell, okay, to make more ATP, to continue to drive more reactions. So this is an endergonic reaction. To go from ADP to ATP is an endergonic reaction. So in order to drive the endergonic reaction, we need an exergonic reaction. So an exergonic reaction that can provide the energy to build up more ATP would be, for example, cellular respiration. So we can couple reactions together, one that releases energy with one that requires energy, to drive reactions in the cell. Here's another way to express a coupled reaction using ATP. Again, this reaction, CD as reactants to products AB, is going to be an endergonic reaction. It's going to require some energy for that to occur. The energy can come from ATP. So as it is converted from ATP to ADP, a little bit of energy is released, and that energy can be used to drive this reaction. And there's lots and lots of reactions in the cell that require ATP to occur. So we've already seen a few of those in previous chapters. For example, the sodium potassium pump that we saw required a little bit of energy to change the shape of that sodium potassium pump in order to change shape and open up and release sodium ions on one side, close up, and then release potassium ions on the other side. In that case, ATP was hydrolyzed to ADP and provided the energy to change that protein's shape. So that would be an example of a coupled reaction that we've already seen. Here's another example of a metabolic reaction, and this is specifically how ATP is used to generate mechanical energy of muscle contraction. So in this case, we have myosin, and remember, myosin is a protein that interacts with the actin cytoskeletal filaments, and in this case, ATP will bind to the myosin protein, and when it does, it causes it to have a certain conformation or shape. The ATP is then hydrolyzed to ADP and phosphate. When the energy is released from that exergonic reaction, it changes the shape of the myosin so that it connects to the actin. This third step here is then ATP and the free phosphate are released from the myosin molecule, and that changes it to a third conformation. And what this does is to move the actin filament here in relation to the myosin fiber, so they move in opposite directions. This is what allows your muscles to contract. So again, the exergonic reaction of ATP to ADP is providing the energy necessary to change the shape of myosin and allow for muscle contraction. A lot of reactions that occur in the cell are endergonic reactions, in that they require an input of energy in order to make the reaction occur. This is what's represented in the graph here. This is the amount of free energy necessary and the progression of the reaction. So you start with reactants and end with products. But in order to get there, either it can happen spontaneously and the level of free energy drops, or more often with endergonic reactions, what we see is that there needs to be an input of energy required to get the reaction going. And that's called the energy of activation. That input of energy allows the reaction to proceed. So where do enzymes, remember enzymes are one of the things we're going to talk about this lecture, where do enzymes come into play with all this stuff about coupled reactions and metabolic reactions? Enzymes decrease the amount of energy necessary to make a reaction occur. So if the blue line represents the normal amount of energy needed to input into a reaction to make it occur, if you have an enzyme present, that can decrease the energy of activation necessary for a reaction to occur. So you can see here the decrease in the energy of activation when an enzyme is present. Metabolic pathways, so this is just a single reaction, but we can also talk about metabolic pathways. Typically what happens is not a single isolated reaction in a cell, but rather a pathway of reactions, where you have A reacts and converts to B, B is converted to C, C is converted to D, D is converted to E, and each one of these reactions, A to B, B to C, C to D, or D to E, would require a different enzyme to facilitate that reaction. So you'd have enzyme A that reacts with A to help change it to B. It decreases the activation energy. A second enzyme, enzyme B, would then react with B to convert to C. Enzyme C converts it to D. Enzyme D converts it to your final product of E. So here in a metabolic pathway, it's a string of these reactions, each one utilizing a different enzyme to facilitate that reaction. Why are so many different enzymes necessary? Enzymes are very specific to the substrate or the reactant that they work with. So enzyme A only works with reactant A. Now enzymes, as we've talked about before, are almost always proteins. However, there are a few examples of what's called a ribozyme, where RNA can act as a catalyst. Catalyst is another word for an enzyme, so there are a few examples where RNA can act as an enzyme in a reaction or a catalyst to catalyze a reaction. But almost always we're talking about proteins. So let's see how enzymes can work with their particular substrates or reactants to form a product. So here we have an enzyme, a protein, and remember proteins have specific shapes. So in this particular shape or conformation of this enzyme, there's what's called an active site. And an active site is specifically where an enzyme is going to bind its substrate or reactant. Okay? It can either be one substrate or two substrates. In this case, we're looking at a synthesis reaction where something is going to be made. So we have two substrates that are going to come together to form a product. The active site is going to be very specific for the shape of the substrates. So these substrates are going to fit very nicely into the active site of the enzyme, and then it's going to form an enzyme-substrate complex. The enzyme itself will help to first of all, it'll help to bring the two substrates together. So they might be floating around in the cell, and they're pretty small in comparison to the big world of the cell. So this enzyme will help bring these two together in close proximity, but also in the right orientation to allow them to react to form a product. So the way that the enzyme in this case reduces the activation energy is because these two substrates have to physically meet and be in the right orientation to form this product. The enzyme will facilitate that through this specific shape of the active site, which will allow it to bind both substrates and bring them in close enough proximity to form the product. So in the case of a synthesis reaction, this is how enzymes would facilitate metabolic reactions. The second example here is a degradation reaction, where we start with a single substrate that then is broken down into multiple products. Again, an enzyme can facilitate this through its active site. It can bind the substrate in the active site to form this enzyme-substrate complex, And in the process, it'll put strain on the bonds that are holding the substrate together, and it can break bonds and form products and release them. So it reduces the activation energy by putting that substrate in the right conformation to allow it to break bonds. An important thing to remember here, you can see the enzyme is released. Enzymes are not used up in the process of the reaction, so they can then go and bind another round of substrates. So they themselves are not utilized or used up in the reaction. There are lots of factors that can affect how well an enzyme works. We'll talk about several of these. The first is pH. Okay, we talked about the pH scale, how it adjusts the relative amount of hydrogen or hydroxide ions to make something either acidic, low on the pH scale, neutral, or more basic. More basic has more hydroxide ions. Acidic has more hydrogen ions. There's different enzymes that are going to work better in different conditions. Okay, if you have an enzyme that needs to work well in your stomach to degrade food particles, you're going to want an enzyme that's going to work well at a low pH. If you have an enzyme in the blood, it needs to be at a more neutral, it needs to work well at a more neutral pH. Hydrogen ions and hydroxide ions in excess can change the shape of proteins. They can protonate, they can deprotonate, they can change the shape of enzymes. If you change the shape of an enzyme, you change the active site, right? The active site is part of its shape. So by changing the pH, you can actually change the active site. If you change the active site, it will no longer bind the substrate as well as it used to. So what happens is it changes how fast the reaction occurs. So most enzymes have a range of pHs at where they work well. For example, pepsin here, this is the rate of the reaction. It has a peak reaction rate in this pH around 2. So it works very well in the environment of the stomach. If you get out of that range, if you get in the pH 5 or 6, it's not going to work well at all. And so we see this kind of bell curve. Peaks up with the rate of reaction at the best pH and then drops again. With trypsin, which might work better at a higher pH, again, you see this bell-shaped curve. Because acids and bases can change the shape of a protein, we see that they can have an effect on how well an enzyme works, because an enzyme, as a protein, needs to have a very specific shape in order to work properly. You change the shape, you change the active site. In a similar manner, temperature can also have an effect on the rate of an enzymatic reaction. And we'll see a very similar bell curve as temperature changes. So here in this case, this reaction is going to work very well right around 40 degrees. 37 degrees is internal body temperature for organisms like humans and other organisms that maintain an internal body temperature. Many of our enzymes will work best at 37 degrees Celsius because that's where our bodies maintain a temperature. What kind of effect does heat have on the shape of a protein? Remember when we did the wire exercise to fold your proteins, and I told you to hold it by the backbone and shake or add heat. What did the heat do? Remember it broke the ionic bonds that were holding amino acids together, and it disrupted hydrophobic interactions, and so it denatured the protein. If a protein is denatured, its shape is changed. If you change the shape of a protein, as an enzyme, it will not work very well because it cannot bind the substrates. So even if you change the temperature a little bit, you're going to have a decrease in the activation, and eventually, if you denature it or you slow the reaction down too much, it won't work well at all. So that's how high temperature can denature proteins and affect the rate of reaction. Low temperature is going to decrease how fast molecules are moving in an environment. So if you slow down how fast molecules are moving, it's less likely that your substrate will bump into the enzyme. Enzyme and substrates have to physically connect and bind at the active site in order for a reaction to take place. So, if you decrease the temperature and the molecules aren't bumping into each other, the reaction is going to slow down. So here, endothermic animals, body temperature promotes the rates of reactions. Ectothermic animals, it sometimes can limit the rate of reactions. They're like these lizards laying out in the sun. If they get too warm, it'll slow down their metabolic rate because their enzymes will slow down. If they get too cold, again, their enzymes will slow down, and so the metabolism will slow down. And there's a good range in which their enzymes will be peak and they'll be most metabolically active. The Siamese cat is actually a very interesting example of the effect of temperature on enzymes. There is an enzyme that helps to make this dark pigment in cats. And in the Siamese cat, that enzyme, as a protein, has a mutation in it that destabilizes it at core body temperature. In other words, the enzyme does not work well at core body temperature, and so it doesn't make the pigment. However, in the extremities, like the face and the ears and the paws, the tips of the paws, that's where the cat is a little bit cooler, and the enzyme works better in that cooler environment due to this mutation. And so there's more pigment made when the areas of the cat are, the parts of the cat that are at a cooler temperature. So this same enzyme in this cat can have different effects in different parts of the body based on temperature. Occasionally, enzymes don't work alone, but require the assistance of a cofactor. Cofactors are inorganic molecules. If they're organic molecules, they're called coenzymes. Both cofactors and coenzymes will fit right inside the active site. So here the cofactor is shown in red, and it fits right into the active site. What it does is to modify the active site just enough to allow a substrate, shown here in yellow, to bind. Without the cofactor, the substrate would not bind very well. So cofactors can help enzymatic reactions proceed by adjusting the shape of the active site so that it binds a specific substrate. Cofactors can be things like copper, zinc, or coenzymes, for example, vitamins, whether or not they're inorganic or organic. And so sometimes we need certain vitamins and ions in relatively low quantities just to facilitate metabolic reactions because they serve as cofactors and coenzymes. In the next two chapters, we'll be talking about two coenzymes, NAD plus and NADP plus, that will serve as cofactors for photosynthesis and cellular respiration. So just something to keep in mind for a future chapter. So cofactors and coenzymes can affect the rate of reaction. So if you don't have a cofactor coenzyme available, the reaction will be limited. But if you have them available, it'll speed up a reaction. Two more things to keep in mind that can affect the rate of a reaction. One is the concentration of the substrate, and the other is the concentration of the enzyme. If you have lots of substrate but very little enzyme, and you add an additional, if you increase the amount of the enzyme, you will speed up the reaction, because then more substrate can be broken down at a faster rate. However, there's a limit to this. Once, and it's typically a one-to-one relationship between enzyme and substrate, once you reach a certain point, all of the active sites on the enzymes will be filled and no more substrate can bind. So you can keep adding enzyme and enzyme, but eventually you'll have too much enzyme for the amount of substrate that you have, and it won't speed up the reaction anymore. So it'll speed it up and then kind of plateau. Remember, enzymes are not used up in the reaction, so it'll keep maintaining, as long as you have substrate to break down, it'll keep maintaining the rate of the reaction. So that's enzyme concentration. The same concept can be applied to substrate concentration. If you have a whole bunch of enzyme and not very much substrate, the reaction won't occur very fast. But if you add lots more substrate, it will speed up the reaction again until all of the active sites on the enzyme are full, and then the reaction rate will plateau as all the active sites are filled. So you can adjust the concentration of the substrate or the enzyme to facilitate how fast a reaction is occurring. The cell can do this because enzymes are proteins. If it wants to make more of an enzyme in order to speed up a reaction, it can. If it wants to limit how much enzyme is present in order to limit how fast that reaction occurs, then it wouldn't make more proteins. It would limit the amount that's made, and the cell has the capability of doing that. So far we've talked about pH, temperature, cofactors, coenzymes, substrate concentration, and enzyme concentration. These are all examples of factors that affect the rate of an enzymatic reaction. Environmental factors, things that the cell can adjust in order to limit or enhance how fast a reaction occurs. The last thing to mention is inhibitors. Inhibitors can have a big effect on enzymatic reactions. There's two different types of inhibitors. Sometimes inhibitors can bind straight to an active site. So what will happen is you have an active site of an enzyme, and you'll have an inhibitor bind, and it'll block where a substrate wants to bind. So it has a similar conformation as a substrate, but it'll prevent the substrate from binding, so it can inhibit the reaction. The other type of inhibitor is what's called an allosteric inhibitor. Allosteric inhibitors will bind to a site other than the active site. The second site is called an allosteric site. Steric just means conformation. So it's another shape, another portion of the enzyme where something can bind to that protein, but it's not going to directly compete with the active site. However, what it does is when it binds, so here's an example of an inhibitor binding to an allosteric site, and when it does that, you'll notice it changes the shape of the active site. It was kind of a semicircle here, and now it's this weird trapezoid shape. If it changes the shape of the active site, it will block the substrate from binding. So it'll inhibit a reaction, but it's called an allosteric inhibitor because it doesn't actively compete with the active site. Cells use allosteric inhibitors very commonly, and we already talked about a metabolic pathway. So here's an example of a metabolic pathway, A to B, B to C, and each step along the way there's an enzyme that facilitates that. What can occur is that the end product of the reaction, so something's finally made in the cell, it itself can go back and bind to the very first enzyme in the reaction, E1 here, and it'll block that pathway from continuing anymore from the very beginning. Why would that be advantageous in a cell? So you have a cell that's busy making product F. Suddenly it has enough product F in the cell. It doesn't need to continue putting energy into making product F anymore. By having product F be the inhibitor to the very beginning of the reaction, once there's enough F in the cell, it can turn off the pathway. So the cell has a way to regulate that pathway quite easily. This is an example of feedback inhibition. The end product is feeding back to the very beginning of the pathway and inhibiting it. Now, once the cell starts using up product F, the levels in the cell will start dropping. And when it does, it'll stop inhibiting enzyme 1, and that pathway can be turned back on again. It's a very easy way for a cell to say, okay, we have enough, let's turn off the pathway. We don't have enough, we need to turn the pathway back on and make more of a product. And so feedback inhibition very commonly occurs in cells and metabolic pathways as a way to self-regulate how much product is being made. Many inhibitors are very advantageous in the cell for this reason. They can feed back and stop a pathway. They can turn on a pathway again when the levels get low. But we also have several examples of inhibitors that are not good. Okay, here is a really, really bad inhibitor. It's called sarin gas. It's a bioweapon used in biowarfare. It inhibits acetylcholine esterase, and this is an enzyme that works at the neuromuscular junction. If you inhibit this enzyme, it really messes up your nerve conduction. The nerve impulses are going to be continually transmitted. You can't turn them off into a relaxed state. A person who encounters sarin gas has difficulty breathing, they vomit, defecate, and eventually it leads to suffocation because your nerves can't stop firing. This picture is showing an example. In 1995, there was a terrorist attack on a Tokyo subway where sarin gas was released and it killed 13 people. It's a really potent enzyme inhibitor. So there are some really bad examples of inhibitors, but many times inhibitors are used in the cell in a very positive way. So by now we've seen how enzymes facilitate metabolic reactions. Metabolic reactions are all about a conversion of energy. Okay, so enzymes can help in the conversion of energy by lowering the activation energy. And we've talked about environmental factors that affect how well enzymes function. So some of the bigger questions of this chapter have been addressed. The last thing that I want to talk about, and this points to where we're headed in the next couple chapters, is the energy organelles and how they play a role in the conversion of energy. The energy organelles in the cell are the chloroplast and the mitochondria. And we've already talked about the structure of these, the fact that chloroplasts help facilitate photosynthesis, while the mitochondria help facilitate cellular respiration. And so as the energy organelles, they help in the conversion of energy. For photosynthesis, it's the conversion of solar energy into the chemical energy of sugars, a stored energy source, both for plants, for animals, for other organisms like bacteria. So these organelles are very important in helping to convert energy from solar energy to sugar. And there are there's metabolic pathways that take place. Basically, photosynthesis is this huge metabolic pathway. And we're going to talk about how enzymes are used in this process in the confines of the chloroplast in order to convert from one energy form to another. In the mitochondria, then, that's where the cell utilizes the sugar and converts it to the energy, specifically the energy of ATP. Okay, so it's chemical energy of sugars converted to chemical energy of ATP. But the ATP is much more usable in the cell because now we have a store of ATP and it can be used to drive other chemical reactions in the cell through these coupled reactions. Things like nerve impulses, protein synthesis, muscle contraction, all of these endergonic reactions that require energy, that energy comes from ATP. The mitochondria, as the powerhouse of the cell, is the organelle responsible for this energy conversion. So again, there's a series of metabolic reactions that occur in this energy organelle to allow for this conversion of energy. And essentially, for living organisms, all energy flows through these two organelles. Solar energy to sugar, sugar to ATP. Some of the products of cellular respiration are recycled, used in photosynthesis. Products of photosynthesis are used and recycled and used in cellular respiration, and so it's a continuous cycle of energy being converted from one form to the other, all flowing through these two energy organelles. With this, we wrap up our discussion on energy conversion and enzymes, and where we're headed in the next two chapters to discuss much more in depth how photosynthesis, the metabolic reactions of photosynthesis, and the metabolic reactions of cellular respiration work. So we'll get many more details about these processes and about how these two organelles facilitate these kinds of reactions. And I hope you'll see these metabolic pathways occurring and how there would be enzymes involved in each step of these pathways.